one day in the mayfly to hundreds of years in humans, the bowhead whales, and even 500 years in the clam. So this raises fascinating questions about what is underlying those amazing differences in lifespan, and also it is important to recognize that most of those species still do age. So what are also the conserved mechanisms throughout species from one day to several hundreds of years that drive the aging process? So in my lab, we're very interested in using animals that are short-lived to identify new genes and new paradigms for longevity. So we use, like many people in the aging field, the worms Brunnet1 elegans, and we've heard several talks about this. We're also developing a new vertebrate systems for aging research, which is the African killifish, Nothobranchus furteri. And then we are also interested, of course, in translating some of the findings that we discover in short-lived organisms, in longer-lived organisms, mammals, and perhaps to identify specific principles that can be learned from long-lived species, in particular the large number of stem cells in those species. But today what I've decided to talk about is our effort to develop the African killifish as a new model systems for aging biology, and also to understand perhaps new findings about what is the evolution of short lifespan versus long lifespans in the wild. Okay, so this project stemmed from the observation that the aging field had really benefited from short-lived model systems like yeast, worms, and flies. But those systems, although they are really awesome to high-throughput screens and etc., they do lack some of the organs and tissues that are very important during mammalian aging. For example, bones, true blood, adaptive immune systems, complex inflammation using complex cells. Now, at the other end of the spectrum, of course, the aging field, at least for experiments, the aging field has used mice, which lives 2.5 years, and zebrafish, which lives 5 years. But those models, as you can see, are long-lived for experimental studies. So it takes, one cannot do screening them, it would be very difficult. And they are actually also quite expensive. This is particularly true for mice. So we felt that there was a niche to fill for a short-lived model system, and this African killifish fills in those characteristics. So what is the African killifish? Well, there are killifiches all over the world, and they tend to live actually in pretty harsh habitat, and pretty much all killifish species are short-lived, about 1 year or so. But the shortest lived of all is this African turquoise killifish, which lives in those ephemeral or transient ponds of water that are only present during the rainy season in Zimbabwe and Mozambique in Africa. And this rainy season lasts about 4 months. And here is the exact same pond during the dry season. So you can see it completely dries up. So the fish has presumably evolved several mechanisms to adapt to this very harsh habitat. One of them is an exceptionally compressed life cycle. It reaches sexual maturity very fast, and it can produce lots of progeny very fast. Another system, and I'll come back to that at the very end of my talk, is this diapause system. Its embryos can enter diapause and can actually survive. That's how they survive, in fact, the complete drought that can last for 1 year and sometimes even 2 years. Now, those fish, even when brought in the lab, so this is our fish room in the lab, in full amount of water, they still recapitulate a short lifespan. So it's so far the shortest lived vertebrate that can reproduce in captivity. It can live 4 to 6 months, even in plentiful amount of water. And despite its very short lifespan, it actually does age. So it doesn't die of sudden death, or it doesn't probably die just when the ponds immediately dry up. It recapitulates, if you wish, a full life cycles and aging, at least in the laboratory. It does display aging phenotypes such as fertility decline, sarcopenia, learning deficit, and neoplastic lesions. And a lot of those have been characterized by Sandro Celerino, who first proposed this model, and also Dario Valenzano, who set up this model, who was a student with Sandro Celerino, and who set up this model in my lab then, as a postdoc. And there also exists different strains with 2 to 3-fold difference in lifespan under set-defined condition in the laboratory, and I'll come back to that also at the end of my talk. Okay, but so what was entirely, so this model is great, so vertebrate model that recapitulates that has a short life cycle and recapitulates signs of vertebrate aging. But it was, at least when we started, entirely missing a set of tools that are needed in order to manipulate it and make it a genetic accessible model organism. So one part that took us a long time actually is as we started working on this, is to develop the tools, basically, develop this animal as a new model for the study of aging and longevity in vertebrates. So that means the genomic, genome editing tools. And the second part, now that we are starting to have some better tools, is to ask fundamental biological questions that can be uniquely asked with this model organisms, because I think it's important not to duplicate the work that has been done, that has been fantastically done in other species. Okay, so the first step with new model systems is to really have a genome and a genome editing. And this task was actually tackled by 3 postdocs in the lab: Itamar Parra for the genome editing, Berenice Benayoun, and Karim Singh for the genome. So this was quite a challenge, because even though those fish are short-lived, they have a very large genome to gigabase. As a comparison, zebrafish is much smaller than humans, of course, of the gigabase. And the other challenge is that 50% of the genome is repeated sequence, so that makes it very hard to assemble using next-generation sequencing. In addition, the closest sequenced fish, which is platyfish and Lidata, are too distant for helping scaffolding the assembly. But nevertheless, we went ahead and sequenced the genome to an 80x coverage, and we have an N50, which means the average size of scaffold of 118. That's not the best type of N50 you can get, but this is a very workable, it's a draft genome still, but it's a very workable genome, and we've worked a lot already with it. And in fact, pretty much all the genes that we've looked for and that are known to be present in fish are in there. We also have done a transcriptome by RNA-seq with 4 different organs, and we started to do epigenome in it with H3 photomutation ChIP-seq, which has really helped us annotate the protein-coding genes, the non-coding RNAs, and etc. in this genome, and also test its quality, and that's why we're pretty confident that it's helpful, and we're happy to share it with the community for people who are interested in looking at their own gene of interest in it, and etc. Okay, so now that we had a genome, the next step was, can we use it now to manipulate this genome? And the advent of CRISPR-Cas9 technology has made it possible to, in fact, manipulate genomes in a much more high-throughput manner than it was possible before. And that's what Itamar decided to do. So taking from this nice wheel from this review by Lopez Martin that Linda Partridge also authors, it's very nice because it gave us a framework with 9 hallmarks of human aging. So Itamar chose genes that not covered entirely this wheel, but that had covered some categories of this wheel. And as an example, I will just summarize his work, because it's now published, but I will just summarize his work with TERT, telomere, the catalytic subunit of telomerase for the telomere attrition, and then I'll say where we are in the other aspects of the mutants. Okay, so as you know, telomerase is the catalytic subunit of telomerase. It's interesting to note that, I think it's serendipitous, but the African killifish has telomere length that are about 8 kilobases, and that's pretty similar to humans, and this was determined by Christophe Englert at Jena. Mouse, in contrast, have very, laboratory mice, has very long telomeres, and that has, in a way, slowed people have been heroic in doing research on telomerase in mice, because we have to intercross mice for 5 generations in order to see a phenotype. As you know, telomere shortening rate has been proposed by Maria Blasco as a biomarker of human aging, and also with Blackburn. And then humans with telomerase deficiency have a disease named dyskeratosis congenita that resemble premature aging. So, with this, Itamar decided to target TERT in telomerase, the catalytic subunit of telomerase in the fish. So this is the CRISPR-Cas9, where it chose the guide RNA, etc. And we were able to get a lot of deletions in TERT, from 3 base pair to 15 base pairs. But the one deletion that we focused on is this deletion of 8 base pair, because it is predicted to give rise to a premature stop codon just upstream of the key domains for TERT, which is the RNA binding domain and the reverse transcriptome domain. So the first question then is, okay, we have a deletion of 8 base pairs, and we were able to get a germline transmission and a nice line of the TERT deletion 8 in this premature stop codon, predicted premature stop codon of TERT. So the next question was, well, is telomerase active as a complex in this fish? We use what's called the trap telomerase repeat assay that measures, basically, we did that with Tibarton, that measures the ability of a tissue, in this case, the liver, to amplify artificial telomeres that are given to it. So you can see that wild-type liver from the fish can amplify artificial, can have a nice telomerase activity. But as far as we could tell, the mutant line that's deficient in TERT had no detectable telomerase activity. Now, what do those fish look like? Well, they're actually pretty normal at first glance. They develop very normally, and they reach adulthood very normally, and they're almost indistinguishable as far as we could tell from the wild-type fish. Those are the mutant, and those are the wild-type. However, when we started to dissect and do like general pathology, we started to see defects. So they have defects in the gut, they have defects in highly replicative type of organs. So they have defects in the gut and in the blood. They do not see defects that we can see in muscles or kidney or heart. But the main defect that they have, and this is also similar to what happens in mammals, is that their main defect is that they have a premature, well, they have a fertility defect, even though they are pretty young actually. And this defect is amplified by aging, but they really have a strong defect in fertility. This is in males, but this defect was also seen in females. So based on all this, this seems to be as far as you can tell, the fastest vertebrate model for TERT deficiency, because it develops defects that are similar actually to the dyskeratosis congenita disease in human, so defect in the blood, in the gut, and in fertility. And develop those defects in the first generation, whereas in mice, as I said, it takes 5 generations of interbreeding to see a phenotype in laboratory mice. And in zebrafish, there is a nice model actually in zebrafish for TERT, but they take longer to display the phenotype because everything in zebrafish is expanded. It takes also 5 years. Okay. So this was the TERT. We chose that because, well, it's the first that went germline, first of all, but also because it exemplified our effort with this wheel. But now we have a series of other stable line, chimera, and genes that are working for where we could test that they were working in the eggs. And we are again happy to share this toolbox of tools and mutants to the community. And if people want to also know how to do that, and it's very efficient, this CRISPR protocols, we're happy to share all the tips for that, because I think like, people can easily do that also in their own lab. Okay, so the questions that we are still asking on that front is obviously now we have a nice model for TERT, so can we use the more high throughput nature of this fish to test a lot of things that wouldn't be possible neither in zebrafish nor in mice. For example, testing a lot of genes and environmental pathways that can rescue TERT deficiency. And obviously we're very interested in testing the importance in lifespan and health span of candidate aging and longevity genes. Genes that, for example, we've discovered in worms in our lab for regulating chromatin and longevity. We would like to know whether or not they have an impact on longevity in vertebrates, and we feel like that's a nice intermediary system for mice. So to summarize, and to also